I'm Xue Han. I'm in biomedical engineering. I run the neuroengineering group in this building. My lab is in this building. So how can we approach clinical problems effectively with animal models? So here is the problem we are focusing on. In particular, this is a therapy called deep brain stimulation. A lot of you are probably familiar because it was discovered in 1987. It was first approved in 1997, and it's been broadly used to treat various types of disorders. Currently, the clinical standard is using about high frequency, 140 hertz, and that's just based on precedent. If you talk to the clinicians, why are you choosing this parameter? Because other people have done it, because this works and others don't work, and it sometimes works. So now the question is, how does it work? And the question is that we don't really know. And how can we approach this problem? And so that's the kind of questions that we are trying to answer in preclinical models. And so when we think about clinical neuromodulation, when we can look in humans is clinical symptoms, but how does the brain respond to these neuromodulation? The way we think about this is I'm going to bring this whole level all the way down to single neurons. Okay, so in the middle, that gray box is the single neuron. The neuron processes information and produces an action potential to transfer this information from input to output. Okay, so there's a lot of natural synaptic inputs, and it generates a decision to make action potential output. So we think about electrical or ultrasonic neuromodulation as just another input. So if we can measure that individual neuron's activity, then we can start to understand how these different clinical neuromodulation works. However, the traditional methods of measuring neuroactivity is using electrode. When we do electrical stimulation, that produce artifacts. So the way we are approaching this is we developed these optical probes so we can translate these membrane potentials into fluorescence change. On the right is a single neuron in a mouse, in a behaving mouse, where the fluorescence change is capturing the membrane potential dynamics. And so now at the bottom is the action potential and the subthreshold membrane voltage dynamics. Now, when we go back to look at electrical neuromodulation, we did this experiment. This is just one of the many examples. We put an electrode next to the neuron. We asked, what kind of frequency would drive the neural response differently? So here, clinically, it's 140. If you give 40 hertz, it doesn't work and sometimes makes the symptom worse. Why? So we measure the neural activity optically while driving or delivering the brain stimulation electrically. So this is no interference. And what we discovered is that membrane potential depolarizes in this scenario, and action potential output is disrupted. When we do, that's with 140, when we do 40 hertz, we found the neurons following this electrical stimulation. So at 40 hertz, it's reliably in training neurons that could produce a lot of set effect, and perhaps that's why it's not working. So with these ideas, we can answer the question. We can't really answer what is the target in the mouse, but we can answer what kind of patterns that we can drive to create informational lesion or to boost informational lesion, or what kind of dose that we can give. Is it high intensity or low intensity? So these are the type of questions that we are investigating. We are interested in deep brain stimulation, ultrasound neuromodulation. If you have clinical problems and you want to know the mechanism, I'd love to talk to you after this over the TED part. And with that, thank you.